This video will be covering the transport of material through the cell membrane. There are 5 basic mechanisms we will be reviewing over: diffusion, osmosis, active transport, filtration, and endocytosis, as well as exocytosis. Remember that the cell membrane is composed of a phospholipid bilayer and that it is semi-permeable, allowing some material to pass through while acting as a barrier to other material. Now, diffusion is the net movement of molecules from a region of high concentration to a region of low concentration. So the molecules are said to move down a concentration gradient. This means that diffusion is a passive process, which means no energy is used to make the molecules move. They have a natural kinetic energy. And so as we look at this, if we were to take this right here, this is an area of high concentration. And so when we look at the molecules moving, as they move out, they are moving from that area of high concentration to this area of low concentration. Just like in this one, as these molecules begin to move from this area of high concentration out, they're moving away from the concentration gradient from an area where we have a lot of molecules to an area where we have very few. Osmosis is the diffusion of water or the movement of water down a concentration gradient from an area of high concentration to an area of low concentration. Therefore, osmosis is a passive process as well and requires no net energy. The net direction of osmosis depends on the solute concentration on both sides of the cell membrane. We can have a hypertonic solution in which we have a higher solute concentration. Now, let's look at what happens in an animal cell versus a plant cell. In an animal cell, this results in water leaving the cell so that the cell would begin to shrivel. The scientific term for shriveling is that the cell crenates. In a plant cell, we have this being the cell wall, this right here being the cell membrane, and so as the water leaves the plant cell, it plasmolyzes, or the water leaves and it pulls the cell membrane away from the cell wall. Here we would see the cell beginning to shrivel up. This is apparent on the side of the road when the ice is put out for snow, and some of that rock salt ice, that rock salt gets on the grass. It can draw this out. You can also make a slide of an onion, and by adding salt water, you can see plasmolysis occurring. We can also have an isotonic solution. An isotonic solution is when we have equal concentrations on both sides of the membrane. So here in an animal cell, we have the same amount of water entering the cell that's leaving, so the cell will appear normal. In a plant cell, we would have the same amount of water entering the cell that's leaving. Again, in this case, the plant cell will not appear normal, but will appear flaccid, and the plant will be droopy. It would need more water and would look like it needed to be watered. A hypotonic solution is where we have a higher concentration of water that enters the cell. So here we have more water entering the cell than leaving. The cell would rupture. The scientific term for rupture is lysis. In a plant cell, we would have more water entering the cell than leaving. The plant cell would be said to be turgid. Now, this is a plant cell that is well watered, and the plant will look very stiff. It will look well watered. There will be no wilting of the plant whatsoever. Here we have osmosis occurring in a red blood cell. And so we can see the three different situations. Here we have a hypertonic solution where we have more water leaving the red blood cell, and so it's beginning to crenate or to shrivel up. Here we have an isotonic solution where we have the same amount of water entering as leaving the cell, so it looks normal. And here we have a hypotonic solution where more water is entering the cell than leaving, and the cell has exploded or lysed open. Dialysis is diffusion through a semi-permeable membrane. So here we have the semi-permeable membrane, and then here we have the material that can pass through the membrane, and we have other material that cannot pass through the membrane. Again, the definition of dialysis is the diffusion of water through a semi-permeable membrane. Active transport is transport that requires energy to move material up and down the concentration gradient. In other words, it requires energy to move material into or out of a cell. So it moves from an area of low concentration to an area of high concentration. Examples of active transport would be the sodium-potassium pump and nerve cells that transport sodium ions to the extracellular environment. Endocytosis is the process by which a cell will take material into the cell. It can take and ingest external fluid or macromolecules into the cell. So this material can be in liquid form or solid form. Therefore, there's two types of endocytosis. There is pinocytosis, known as cell drinking or bringing liquid into the cell, and there is phagocytosis or cell eating, which is bringing solid into the cell. Phagocytosis is the term that is most familiar to most biology students, as we hear that white blood cells phagocytize various foreign bodies that infect the body. Exocytosis is the opposite of endocytosis, so this is the removal of material from a cell. This can be in the form of the mucus glands secreting mucus and it leaving the cell, or saliva leaving the cell, or tears leaving the cell, or pus leaving an area. But anytime material leaves a cell, this is known as exocytosis. Filtration is when molecules pass through a membrane by physical force during filtration. A good example of filtration would be with the urinary system and how the kidneys filter out with blood pressure the different components and separate the waste out of the blood.